Hello, my name is Ana Carolina Borges Campos, and today I represent my work titled Surface Modulation with Microtopographies for Directing Chondrocyte Morphology and Phenotype. This project is a collaboration between the Federal University of Rio de Janeiro in Brazil and the Institute of Material Science of Mulhouse in France. And our work is focused mainly on cartilage cells called chondrocytes. These cells are highly sensitive to the microenvironment and they respond to mechanical and physical cues from the extracellular matrix through mechanosensing and mechanotransduction. And an important aspect is that chondrocytes morphology is directly associated with their phenotype. So rounded chondrocytes tend to maintain a healthy chondrogenic phenotype, while flattened cells usually differentiate into fibrotic or hypertrophic states, which compromises cartilage function. This is particularly relevant in therapeutic strategies for articular diseases that require cell expansion in vitro. So during passaging, the chondrocyte will progressively lose their phenotype, which will affect the cartilage production after reimplantation. So, although many studies have investigated the effects of substrate stiffness on chondrocyte behavior, the influence of surface topography is still not fully understood. So, the objective of this work is to produce biocompatible surfaces with microtopographical designs and to elucidate how they will regulate chondrocyte cytoskeletal organization, proliferation, and extracellular matrix deposition. So, to fabricate the surfaces, we first designed microtopographical patterns using the Blender software, combining with grayscale lithography and perspective rendering in the Z-axis. And we created two main categories of structures, the anisotropic grooves, which are expected to induce cell elongation, and the isotropic hemispheres that are expected to promote rounded cell morphology. So after that, we fabricate molds using a photosensitive resin, and this process involves spin coating and soft baking steps for resin deposition, UV exposure for pattern printing, and chemical development for pattern revelation. After obtaining our master molds, we replicate them in PDMS through casting and thermal curing, generating flexible and biocompatible substrates. And finally, the PDMS structures are functionalized using fibronectin coating for cell adhesion and a UV bath for sterilization and activation. To validate the structures, we characterize the surfaces using stillus perfilometry and confocal microscopy. So the perfilometry data first confirmed that the resin thickness we had were ranging from 10 to 30 micrometers using different parameters of speed in a 30-second rotation protocol were correct and reproducible. And we then confirmed the reproducibility and dimensions of the printed microstructures that we intended to make. And we also have confocal microscopy images that demonstrate the successful fabrication of both grooves and hemisphere geometries with controlled organization and spacing. So based on previous observations from the group, we expect that the groove structures will guide cytoskeletal alignment and induce elongated cell morphology. And in contrast, the hemisphere structures that will become wells after PDMS replication are expected to maintain a rounded morphology associated with the preservation of the chondrogenic phenotype. Primarily, articular chondrocytes will be cultured on the surfaces, and they will be compared to flat controls regarding morphology, proliferation, viability, and the expression of chondrogenic fibrotic and hypertrophic markers. So in conclusion, we can say that we successfully fabricated reproducible microtopographical PDMS surfaces with controlled isotropic and anisotropic geometries. And these platforms will help us better understand how physical surface cues can influence chondrocyte behavior and phenotype maintenance. And we believe that by identifying topographical conditions that best preserve the chondrogenic phenotype, we can help reduce chondrocytes the differentiation during in vitro expansion and maybe contribute to improved cartilage tissue engineering strategies. So I would like to thank CNPq for funding this project in this collaboration between Brazil and France. And thank you all very much for your attention. I'll be happy to take any questions you might have.